Welcome to the Deep Dive, where we take a stack of sources and really pull out the most potent, maybe surprising, and definitely useful knowledge just for you. Today, we're plunging into a world where these tiny, often unseen creatures wield just immense power over some of the planet's largest, most iconic animals. I mean, how can something as minuscule as a burrowing might cause debilitating skin disease in a camel? Or a tick, barely bigger than a poppy seed, connect a wild deer to serious human illness? It's quite a biological puzzle, isn't it, with really profound consequences.
It truly is, yeah. This deep dive is all about ectoparasites, those fascinating, sometimes troubling organisms that live on the outside of an animal.
Right, the external ones.
Exactly. And our insights today, they come from a deep comparative analysis of ectoparasitism in camelids, camels, llamas, alpacas, and cervids, so deer, elk, moose. We're looking at everything, the specific diseases they cause, how they spread, epidemiology, and the very different ways we approach managing them.
Okay, so our mission today is really to pull back the curtain on this complex biological dance. We want you to understand not just what these parasites are, but their profound impact, their really surprising differences in how they interact with camels versus deer, and crucially, how we approach managing them, from the health of a single animal all the way up to vast ecosystems and even public health. So by the end, you should have a much clearer picture of this unseen world and its very real-world implications. Let's dig in.
Absolutely. So first things first, what exactly are ectoparasites? Well, essentially, they're organisms living on the external surfaces of a host, and they're getting their nourishment from that host. We're primarily talking about arthropods here, and for our purposes, they split nicely into two main classes. Arachnida, that's your ticks and mites,
Okay.
and insecta, which covers lice, fleas, keds, and various types of flies. And they feed in different ways, some on blood, others on tissue fluids, lymph, or even like skin debris, keratinous stuff.
And when an animal has them, we just call it an infestation.
That's the term, yeah,
Right.
infestation.
Got it. And their life cycles. You mentioned those are diverse. That sounds like it complicates things quite a bit.
Oh, hugely. They are incredibly varied. Some, like many lice species, are permanent parasites. They live their entire life cycle right there on one host animal.
Okay, simple enough.
Relatively. But then others, like many biting flies, are temporary. They just, pop in for a meal periodically.
Hit and run feeders.
Sort of, yeah. And then you get the real strategists. Like many tick species, they have these complex multi-host life cycles. They actually need different hosts for their larval stage, their nymphal stage, and their adult stage.
Wow, okay. That's intricate.
It is. And this incredible diversity in their life strategies, it profoundly influences how they spread their epidemiology, what kind of damage or pathology they cause, and of course, the best ways to try and control them.
So it sounds like it's a double whammy for the host animal, isn't it?
Yeah.
There are the direct impacts just from them being there, and then these more insidious indirect impacts where they're spreading other diseases.
Precisely. That's a great way to put it. Let's start with the direct impacts. When you have blood-feeding parasites, think sucking lice, ticks, biting flies in heavy numbers, they can actually consume significant amounts of blood. This can lead directly to anemia, weakness, and just generally poor body condition.
Okay, that makes sense. Physical drain.
Yeah. And then there's the direct tissue damage. The burrowing activity of mites, like the mange mites, Sarcoptes, and the feeding action of ticks and flies, it causes direct mechanical damage to the skin. This results in inflammation, intense itching, we call it pruritus in vet terms, and pain.
Which must drive the animals crazy.
Absolutely. So the animal, trying to get relief, will constantly rub, scratch, or bite at those areas. That causes self-wounding. All of this leads to dermatitis, hair loss, alopecia, and the formation of crusts and scabs. And that can severely damage hides and fleece, which is a major economic loss, especially in animals like camels.
Right. And beyond just the physical damage, I can only imagine the sheer stress, the toll that constant irritation must take on their overall well-being.
It's huge. It absolutely is. The chronic irritation, the annoyance, the pain, it leads to a state of chronic stress. And this stress, it disrupts normal feeding behavior, reduces how much they eat, and diverts energy away from growth and production towards just trying to cope immune responses, tissue repair.
So they just don't thrive.
Exactly. Infested animals show poor weight gain, reduced milk yield in females, decreased reproductive performance, sometimes even death. And there's another direct hit, toxicosis and allergic reactions. Saliva from some ectoparasites, particularly ticks, can contain toxins. Some can even cause paralysis, tick paralysis. Plus, repeated exposure to the parasite's antigens can trigger hypersensitivity or allergic reactions in the host, leading to really severe inflammatory skin disease.
Okay, so that's the direct punch. Pretty nasty.
Yeah.
But you mentioned the indirect impacts are often even more devastating. This is where it becomes more than just irritation, right? This is the public health angle.
It really is. This is their role as vectors. It's crucial. Ectoparasites are absolutely critical in transmitting numerous protozoan, bacterial, rickettsial, and viral diseases. For instance, biting flies are the primary mechanical vectors of Trypanosoma evansi, which causes sura in camels. We'll get into what mechanical means. Meanwhile, ticks are biological vectors for the agents of Lyme disease, anaplasmosis, babesiosis in deer.
Okay, mechanical versus biological. That sounds important.
Very. This vectorial capacity, it transforms ectoparasites from, just an animal welfare issue into a major concern for the economics of the animal industry and, crucially, for public health.
So what actually makes one animal more susceptible than another? Is it just bad luck? Or are there specific factors at play?
Oh, it's far from random. It's a really complex interplay of host factors, parasite factors, and the environment. Host-related factors are critical in determining susceptibility. Studies consistently show things like age, sex, nutritional status, body condition, immune competence. They all influence parasite loads.
Interesting. So like older or weaker animals are hit harder?
Generally, yes. Young animals with developing immune systems and older or malnourished individuals with weaker immunity, they're often much more vulnerable to severe infestations and the diseases they carry. And an intriguing detail, males are often more heavily parasitized than females.
Really? Why is that?
Well, it's potentially linked to hormonal differences, but also maybe behavioral patterns, like males roaming wider territories, which increases their exposure.
Fascinating. And the environment must be huge too, right?
Yeah.
Where they live dictates what they encounter.
Absolutely critical. Environmental and ecological factors really shape these host-parasite interactions. Seasonality is a big one. Many ectoparasite populations, especially flies and ticks, peak during warmer, more humid months that favor their development and activity.
Makes sense. Summer pests.
Exactly. And habitat structure matters too. Ticks, for example, they thrive in specific microclimates. Think dense vegetation, leaf litter, places that offer protection from drying out.
Okay. And finally, host specificity. Do these parasites just jump on anything that moves, or are they picky eaters?
That's a key point, and it varies. Some parasites, like many species of lice, are highly host-specific. They can only survive and reproduce on one or maybe a few closely related host species.
Okay, loyalists.
Pretty much. Others, though, like certain ticks, are generalists. They'll happily feed on a wide range of hosts. And this distinction has huge implications for disease transmission and control. Host-specific parasites. Often you can manage them by treating just the target host population. But generalist parasites, that requires a much broader ecosystem-based approach. You might need to manage multiple wildlife reservoirs, the environment itself.
Right, much more complicated.
It is. So the key takeaway here is that high infestation rates and disease outbreaks happen at the intersection of a susceptible host, a favorable environment, and peak parasite activity. It really underscores why you need an integrated approach to management, something that goes way beyond just reaching for the chemicals.
Okay, that's a great overview. Let's pivot now and look specifically at camelids. We're talking dromedary, Bactrian camels, plus llamas and alpacas.
Yeah.
What are their biggest ectoparasite headaches?
Right. For camelids, the number one dermatological threat, arguably the most serious and debilitating ectoparasitic disease they face, is sarcoptic mange.
Yeah.
It's caused by a tiny burrowing mite, Sarcoptes scabivar cameli.
Sarcoptic mange.
Okay. And imagine this. Something so tiny, yet it's often ranked as the second most economically damaging disease for camels right after sura, which is trypanosomosis.
Wow. Second only to sura. That's huge.
It is. It's not just an itch. It's a highly contagious crisis found globally wherever camels are raised.
So how does it spread? And what does that actually do to the poor camels?
Well, the mite is an obligate parasite. It has to live on the host.
Yeah.
And it spends its entire life cycle in the skin. It literally tunnels into the skin to lay its eggs.
Oh, okay.
So transmission happens really easily through direct physical contact between animals. In managed herds, this is made worse by close confinement, gathering at watering holes, feeding areas.
Shared spaces.
Exactly. And indirect transmission via contaminated objects, we call them fomites, things like harnesses, bedding, rubbing posts, that's also significant. While all ages can get it, it's often much more severe in young animals or those in poor condition. And interestingly, outbreaks often happen in the colder months.
Colder. Why then?
Likely because longer hair coats provide a better microenvironment for the mites, and also because animals tend to huddle together more for warmth, increasing contact.
Right, right. And the poor camels must be just miserable, constantly irritated.
Utterly miserable.
Hmm?
The main problem, the pathology, is driven by the female mites burrowing to lay eggs. This activity, plus mite feces and saliva, triggers an intense hypersensitivity reaction in the host. That means severe, persistent itching pruritus.
So they just can't stop scratching.
They can't. They become restless, rub, scratch, bite at the infested areas constantly. It usually starts with small red bumps, often on the inner thighs, groin area, neck, shoulders. But these quickly progress. You see hair loss, weeping skin, and then these thick, gray, corrugated crusts and scabs form.
Oh, wow.
Yeah, and in chronic cases, the skin becomes markedly thickened and wrinkled. We call that lichenification. This constant irritation disrupts grazing, leads to loss of appetite, weight loss, emaciation, a big drop in milk production. Plus, the self-trauma creates open wounds, which get secondary bacterial infections or even fly-strike myiasis. And it's zoonotic, too.
It can pass to humans.
It can cause a transient but really intensely itchy dermatitis in people who have close contact with infested camels. We call it pseudoscabies.
Good to know. So
Yeah.
how do you even diagnose something so tiny and hidden deep in the skin?
Well, diagnosis often starts with those characteristic clinical signs, the intense itching, the progressive skin lesions. But for confirmation, you really need microscopic identification of the mites, their eggs, or even fecal pellets. You do this by taking deep skin scrapings, ideally from the edge of an active lesion.
Deep scrapings. Sounds unpleasant.
It can be. You treat the sample with 10% potassium hydroxide KOH, which dissolves skin debris and makes it easier to see the mites under the microscope. But honestly, in chronic cases or if the mite numbers are low, it can be difficult to actually find them. You can get false negatives.
Right. In treatment, if they're burrowed in, topical stuff might not reach them easily.
Exactly. Topical acaricides like sprays or dips can work, but they usually need multiple applications and really vigorous scrubbing to penetrate. So systemic insecticides, drugs that work throughout the body, have become the treatment of choice, particularly the macrocyclic lactones like ivermectin and doramectin.
Injections.
Usually, yeah. Subcutaneous injection is often more practical for large herds, and some comparative studies suggest that doramectin might actually be a bit better, maybe superior efficacy, longer duration of action, clearing infestations faster, and giving longer protection.
Interesting. Doramectin.
But successful control isn't just about drugs. It needs an integrated approach. You have to treat all animals in the herd at the same time. You need to decontaminate equipment, bedding, harnesses, and improving overall animal nutrition and husbandry helps boost their own immunity.
Makes sense. A multi-pronged attack. What about ticks on camels? I imagine those are easier to spot.
They usually are, yes. Ticks are actually the most common ectoparasites found on camels, especially in those arid and semi-arid regions. Camels can host over 20 species of hard ticks, the exuded ticks, but the genus Hyaloma is really the most important one.
Hyaloma.
And the most prevalent species on camels is Hyaloma dromedari. It's desert-adapted, found everywhere camels are. It's typically a two-host tick.
Two-host, meaning?
Meaning it feeds on one host as a larva and nymph, drops off, molts, and then finds a second host as an adult. And heavy tick infestations cause significant direct damage. The attachment, the feeding, it leads to irritation, inflammation, blood loss.
Anemia again.
Yep, can cause anemia, poor body condition, reduced productivity. It can even kill young calves if the infestation is severe enough. Plus, the bite wounds are entry points for secondary bacterial infections and fly stroke.
And do these hyaloma ticks transmit diseases in camels?
Yes, they are vectors, although perhaps their role is less clearly defined than in, say, cattle, but it's still significant. Hyaloma species are known transmitters of the virus causing Crimean Congo hemorrhagic fever, CCHF. That's a severe zoonotic disease in humans.
CCHF. Okay, serious stuff.
Yeah, and high seroprevalence, meaning antibodies are found in some camel populations, suggests they play a role in maintaining that virus cycle. These ticks on camels have also been found carrying bacteria like Rickettsia epsilomani, which causes a type of spotted fever. Now, camels can be infected with protozoans like Filaria, Babesia, Anaplasma, which cause major diseases in other ruminants. but their role as natural reservoirs for these seems less clear, and clinical disease from them isn't commonly reported in camels.
Interesting distinction. What about lice? Are they a big issue?
Louse infestations, peticulations are common, yeah, though maybe often considered less severe than mange or heavy tick burdens. The main one affecting camels is a sucking louse, Microtheraceous camelidae. As the name suggests, it feeds on blood.
Sucking louse. Got it.
Lice are usually highly host-specific, and their permanent ectoparasite's whole life cycle happens on the camel. The female glues her eggs, the nits, to the hair shafts. The whole cycle takes about 3 to 4 weeks.
And the impact? Just itchy.
Light infestations might not cause much trouble, but heavy ones. Definitely significant irritation, restlessness, intense itching. Constant irritation disrupts feeding and rest, leads to poor condition. And because of their blood feeders, large numbers can cause anemia, especially in young or weak animals. Plus, the scratching damages the hide.
Right. And flies. I imagine flies are just a nightmare for any large animal, but maybe especially camels in hot climates.
Oh, absolutely. Several types of biting flies are major pests. You've got stable flies, stomoxys, horseflies, tabanis, and camelkids, Hippobosca camelina. In these groups, both males and females feed on blood, and their bites are painful.
Ouch.
Yeah. So the direct impact is blood loss, cane, severe annoyance. The camels spend time on defensive behaviors, swatting, running, instead of grazing, which hits productivity. But the most critical role of these biting flies in camel health is their function as mechanical vectors of Trypanosoma evansi.
Ah, Sura again. So the flies physically carry it.
Exactly. They pick up the parasite feeding on an infected camel, and if that meal is interrupted, they can transmit it to a healthy animal on their contaminated mouth parts when they bite again shortly after. No development in the fly, just physical transfer.
Okay. And what about that nasal bot fly you mentioned earlier? Cephalopina titillator. That sounds particularly nasty.
It really does, doesn't it? The larvae of this fly are obligate parasites causing what are called nasopharyngeal myiasis. It's an infestation of the nasal passages and throat area. The adult female fly, which doesn't feed, actually deposits live first-stage larvae into the camel's nostril.
Deposits live larvae.
Yeah. These tiny larvae then migrate up into the nasal passages and the pharyngeal pouches at the back of the throat. There, they attach and develop through 3 larval stages, or instars. They can get quite large, these spiny larvae. Their presence causes intense irritation. You see nasal discharge, sneezing, snorting, head shaking.
Trying to get them out.
Exactly. And in heavy infestations, they can physically block the airways. That causes respiratory distress, difficulty swallowing. This can lead to anorexia, weight loss, and in rare cases, actually death from suffocation or from secondary bacterial infections leading to meningitis.
Goodness, that's quite a list of afflictions for camels.
Yeah.
Now let's pivot to the other side of our comparison, cervids. So deer, elk, moose. How do their ectoparasite challenges stack up, especially compared to camels?
Okay, shifting gears to cervids. Here, the story is often dominated, especially in North America, by one particular player, the black-legged tick, Ixodes scapularis, often called the deer tick.
The infamous deer tick.
Indeed, and it's arguably the most medically important ectoparasite of wildlife in North America. Why? Because it's the primary vector for a whole host of pathogens causing serious human diseases.
Right. This is the Lyme disease connection everyone knows about. And you mentioned it's expanding its range.
Exactly. Scapularis is a 3-host tick. Larva, nymph, adult. Each stage usually needs a different host. The whole cycle typically takes 2 years. Larvae and nymphs feed on a wide range of small to medium-sized animals, rodents, birds. White-footed mice are particularly important as they're often the main reservoirs for many of the pathogens.
So the mice keep the diseases going.
In many cases, yes. But the adult ticks have a strong preference for larger mammals. And white-tailed deer are the key definitive host. Deer are absolutely essential for the tick's reproductive success. A single deer can host hundreds of adult ticks. They mate on the deer, the female engorges on blood, drops off, and lays thousands of eggs.
Thousands. Okay, so deer are tick factories, basically.
In a way, yes.
Yeah.
They're crucial for maintaining high tick populations. And yes, the geographic range of this tick has expanded dramatically over recent decades, particularly in the U.S. Northeast, Mid-Atlantic, Upper Midwest. And that expansion directly correlates with the rising incidence of the diseases it transmits.
So it's not just Lyme disease this tick carries. Sounds like a major public health issue.
It really is. The public health burden is immense. Scapularis is a competent biological vector, we'll unpack that term, for at least 7 human pathogens. Bacteria like Borrelia burgdorferi, the cause of Lyme disease, Anaplasma Phagocytophilum, causing anaplasmosis, Borrelia miyamotoi, causing a relapsing fever, Ehrlichia miris occlurensis. Then there's the protozoan Babesia microti, causing babesiosis, and even viruses like Powassan virus, which is rare but causes severe neuroinvasive disease.
That's a scary list.
Yeah.
And Lyme is the most common vector-borne disease in the U.S., right?
It is, and its incidence is directly tied to the density of infected nymphal ticks. The nymphs are tiny, hard to spot, and they're most active in summer when people are outdoors. That's why they cause most human infections.
Okay, so deer are central to the tick's life cycle. But do the deer themselves get Lyme disease or these other illnesses?
That's a really critical point. While deer are absolutely central to the tick's life cycle, they are not competent reservoirs for the Lyme disease Spirochete Borrelia burgdorferi. Meaning the bacteria don't replicate well in deer, and deer don't really get sick from it. They don't maintain the infection in a way that readily passes it back to ticks. However, because they are the primary host for the adult ticks, they act as critical amplifiers of the tick population itself. So high deer densities are very strongly correlated with high tick densities and therefore increased human risk for all these tick-borne diseases.
Got it. So more deer equals more ticks equals more risk. What about mites and lice in deer? Are they significant problems like the mange in camels?
Well, less significant from a direct public health view compared to ticks, but yes, mites and lice can cause severe direct problems for deer populations, sometimes leading to significant illness and death. Sarcoptic mange can affect deer intense itching, hair loss, crusty skin, similar to camels. Dimodectic mange from Demodex mites is also frequently reported in deer, elk, mule deer. Yeah, these mites are normally found in hair follicles, usually harmless, but in animals that are stressed, malnourished, or immunocompromised, the mite populations can explode. That leads to demodicosis hair loss, dry, thickened, flaky skin. Usually not fatal itself, but severe cases can mess up their ability to regulate body temperature.
Okay. And I've heard about something called hair loss syndrome in deer out west. What's the story there?
Yes, hair loss syndrome, or HLS. That's a particularly severe condition seen in black-tailed deer and mule deer, primarily in the western U.S. And interestingly, it's not caused by native lice. It's caused by heavy infestations of exotic chewing lice.
Exotic lice?
From where? Species like Demolinia and Bovicola tibialis. Their natural hosts are non-native cervids like fallow deer or Sitka deer that were introduced. In their original hosts, these lice don't cause much trouble, but in North American deer, the infestation triggers a severe hypersensitivity reaction, an allergic response.
Ah, so the deer's immune system overreacts.
Exactly. This leads to intense irritation, excessive grooming. The deer literally break off their coarse outer guard hairs, exposing the lighter-colored underfur. You see these characteristic patches of yellow or white fur, often along the flanks and back. It's worst in late winter, early spring, when the animals are already nutritionally stressed. And losing that insulating outer coat, it can lead directly to hypothermia and death, especially for fawns. It's a really stark example of how an invasive parasite can devastate a native, naive host population.
Wow, that's incredible, just from a louse. Are there other winged parasites besides the biting flies we associate with livestock that bother deer significantly?
There are. One notable one is the deer kate, Lepoptena cervi. It's another invasive species, originally from Europe and Asia, now established in parts of North America.
A kid. Is that like a fly?
It is. It's a blood-sucking parasitic fly. The winged adult locates a host moose, elk, deer, probably by sensing movement and dark shapes.
Yeah.
But here's the unique part. Once it lands on the host, it sheds its wings.
It sheds its wings.
Yep. Becomes a permanent, flightless ectoparasite living in the animal's coat, taking repeated blood meals.
That is bizarre and kind of creepy. A flightless fly living on you.
It's a very effective adaptation. And in newly invaded areas, they can reach incredibly high densities. There are reports of over 17,000 keds found on a single moose.
17,000. Good grief.
Yeah. The direct health impacts are still debated, maybe linked to hair loss, poor condition. But they are known vectors for the bacterium Bartonella schoenbokensis. And here's an intriguing detail for anyone who spends time in the woods in affected areas. They're a considerable nuisance to humans. They frequently land on people and will bite.
Oh, great. Something else to watch out for. What about nasal bots? You mentioned them in camels. Do deer get those too?
They do. Very similar story, different genus. Larvae of several species of Cephanemia botflies are obligate endoparasites of the nasopharyngeal region in cervids. Again, the adult female fly deposits live first-stage larvae near the nostrils. The larvae migrate up into the nasal passages and the retropharyngeal pouches, attach and develop over months, feeding on mucus.
Same symptoms. Sneezing, discharge.
Mostly, yes. Most infestations are probably subclinical, causing little harm, but you can see irritation, sneezing, snorting, nasal discharge as the deer tries to expel them. In very heavy infestations, same potential problem as in camels. The larvae can physically obstruct airways, causing respiratory distress, difficulty swallowing, and rarely death by suffocation. And pathology studies show they cause significant inflammation and tissue damage in the nasal area, which could potentially impair the deer's crucial sense of smell.
Impairing smell must be a big deal for a wild animal.
Huge deal for predator detection, finding food, finding mates. Yeah, once the larvae mature, they get sneezed out, fall to the ground, pupate in the soil, and the cycle continues.
Okay, this comparative look is fascinating. And you hinted at this earlier. This seems to be where the grand strategy of these parasites really diverges. The way they transmit diseases in camels versus deer sounds fundamentally different.
It really is. This is perhaps the most significant difference between the ectoparasite situations in camels and deer. It's all about their vectorial capacity and the nature of the diseases they transmit. It creates two vastly different epidemiological landscapes and different public health implications.
Okay, lay it out for us. Camelids first.
Right. For camelids, the most devastating vector-borne disease, as we mentioned, is Surra, caused by Trypanosoma evansi. And critically, this pathogen is transmitted mechanically by large biting flies, tabanids, horseflies, and musids, like spable flies.
Mechanical transmission. Remind us what that means exactly. The pathogen doesn't change inside the fly.
Correct. It's a relatively simple physical transfer. The fly feeds on an infected camel, gets the trypanosome on its mouth parts. If it's interrupted and quickly bites another camel, it just physically transfers the pathogen, like using a dirty needle. The pathogen does not develop or replicate within the fly vector. It's just hitching a ride on the mouth parts for a short time. And while camels do get ticks that can carry things like CCHF virus, the overall incidence of clinical tick-borne diseases like filariosis or babesiosis seems substantially lower in camels compared to, say, cattle.
Okay, so mechanical transmission by flies is the big story for camels. How does that contrast with deer?
It's a completely different picture. The disease landscape for cervids, particularly in North America, is dominated by pathogens that undergo biological transmission.
Biological transmission. Okay, how does that work?
This is much more complex. Here, the vector, primarily Echinococcus scapularis, ingests the pathogen when it feeds on an infected reservoir host, often that white-footed mouse we talked about. The pathogen then has to replicate, develop, and/or migrate within the tick's body. It might change forms, multiply, eventually reaching the tick's salivary glands. Only then can it be transmitted to a new host when the tick feeds again. The tick is acting like a living incubator and syringe.
I see. The tick is an essential part of the pathogen's life cycle.
Exactly. This creates what we call an enzootic cycle, a disease cycle that's constantly maintained in nature within a specific animal population and geographic area. It involves multiple players: the tick vector, the reservoir hosts that maintain the pathogen, like mice, and amplification hosts, like deer, which amplify the tick population. The result is this persistent landscape-level risk for a whole suite of serious zoonotic diseases: Lyme, anaplasmosis, babesiosis, powassan.
So the implications for trying to control these diseases must be worlds apart, depending on whether it's mechanical or biological transmission.
Totally different challenges. Managing Surra in camels. The focus is largely on controlling the biting fly populations and treating infected camels promptly to reduce the source of infection for the flies. But managing Lyme and other tick-borne diseases associated with deer, that requires a much more complex, multifaceted strategy aimed at disrupting that intricate and zootic cycle. You might try to reduce tick populations directly, manage the reservoir host populations, like rodents, manage the tick amplification hosts, the deer, and focus heavily on preventing tick bites in humans through education and personal protection.
It really highlights how understanding the transmission mechanism is key to control. Before we get into diagnostics, there's one more thing you mentioned that sounded important for both groups: dermatophilosis. Why is that relevant here?
Yes, dermatophilosis, also known commonly as rain rot or sometimes cutaneous streptothrombosis. This is crucial for accurate diagnosis in both camels and deer. It's caused by a bacterium, Dermatophilus congolensis. And the key thing is it presents with clinical signs that can look very similar to parasitic skin diseases.
Like mange.
Exactly. You see exudative or weeping dermatitis, crust and scab formation, hair loss. It can really mimic mange or severe lice infestations. What's fascinating is the synergistic relationship it often has with parasites.
Synergistic. They help each other out.
In a way, D. congolensis needs two things to really take hold: prolonged moisture on the skin and some kind of break in the skin's integrity. Moisture activates dormant bacterial spores, and they invade the epidermis through minor cuts, abrasions, punctures, lesions that are very often caused by the bites of ectoparasites like ticks and flies.
Ah, so the parasites open the door for the bacteria.
Precisely. And conversely, the inflamed weeping skin caused by a D. congolensis infection can actually attract more flies, potentially worsening the situation or facilitating fly strike. Dermatophillosis is reported in both camels and deer, especially during wet, humid conditions.
So you absolutely can't just assume it's parasites if you see crusty skin. You need to be a diagnostic detective.
You really do. A definitive diagnosis of skin disease in these animals needs laboratory confirmation. Clinical signs can be misleading. Skin scrapings and impression smears should be examined microscopically not only for mites, but also for the characteristic tram track or railroad track appearance of the D. congolensis bacterial filaments. If you miss one or the other, your treatment is likely to fail. It really underscores the need for a thorough diagnostic workup in any case of dermatitis in these animals.
Okay, that makes perfect sense. So let's talk diagnostics. How do you figure out what's going on, especially given the huge difference between a camel in a managed herd and a wild deer roaming the forest? Sounds like two completely different ballgames.
They are fundamentally different. The diagnostic strategies reflect that stark contrast between managing domestic livestock versus monitoring free-ranging wildlife. For camels and other managed camelids, the focus is on the individual animal and the herd. You can handle them, examine them closely.
Hands-on approach.
Right. So it starts with a thorough physical exam. Visual inspection for parasites, looking for those characteristic skin lesions, hair loss, redness, crusting. You'd palpate the skin, check lymph nodes, assess body condition score. For sarcoptic mange, as we said, the gold standard is microscopic examination of deep skin scrapings treated with KOH. Any ticks or lice you find can be collected and identified morphologically based on their features. Blood work is also key. Blood smears are routine for looking for blood-borne pathogens, especially Trypanosoma avance for Sura. Hematology can show anemia, and you can use more advanced lab techniques too. A lysa test can detect antibodies showing exposure. PCR is highly sensitive for detecting pathogen DNA, either in blood or even within the vectors you collect, confirming active infection.
Okay, a whole toolkit for the managed animals. But for wild deer, you obviously can't just walk up and do a skin scraping or draw blood most of the time. So how on earth do you gather data on parasites across an entire population?
Not easily. That's the challenge. Monitoring ectoparasites and their diseases in wild deer populations involves significant logistical hurdles. So we often rely more on indirect methods and opportunistic sampling. Passive surveillance is a major component. This relies on reports from the public hunters, landowners, hikers who observe and report sick or dead deer.
So people calling in if they see a deer that looks really mangy or has lost all its hair.
Exactly. Conditions with dramatic signs like severe hair loss syndrome or advanced mange are most likely to get reported this way. It's useful for spotting unusual events or new outbreaks, but it's definitely biased towards the obvious cases and where people are looking.
Right, so how do you get more systematic data, not just waiting for calls?
That requires active surveillance. One primary method is examining hunter-harvested deer. Biologists can set up at check stations or work with meat processors to collect ectoparasites, tissue samples, blood samples from a large number of animals across a wide area during hunting season. This provides invaluable data for mapping where parasites are, how common they are, and screening for various pathogens.
Clever use of the hunting season. What else?
Another key active method is environmental sampling, especially for ticks. This involves researchers literally dragging or flagging vegetation with a cloth, usually white flannel, to collect host-seeking ticks. You can then count the ticks and test them for pathogens. This gives you a direct measure of disease risk to humans and animals in a specific area.
Dragging a cloth through the woods. Simple but effective.
It is. And finally, necropsy, the post-mortem examination of deer found dead or those euthanized due to severe illness, is absolutely critical. Necropsy allows for a full assessment. You can see gross pathology, collect a wide range of tissues for testing, and recover internal parasites, like those nasal bots that you'd never see on a live animal exam. It's a cornerstone of wildlife disease surveillance.
Okay, so vastly different diagnostic approaches, which leads us to management. Given these different contexts, managed camels versus wild deer, different transmission modes, different diagnostic tools, the management strategies must also be fundamentally distinct.
They are, completely. It boils down to this. Managing ectoparasites in camels is primarily a veterinary and animal husbandry challenge focused on the individual and the herd. Managing them in deer is largely an ecological and public health challenge focused on populations and ecosystems.
Okay, let's break that down. How do you manage them in camels? Sounds like drugs are key.
Pharmacological control using chemical agents is definitely the cornerstone for managed camelids. Those systemic macrocyclic lactones we talked about, ivermectin and doramectin, are highly effective against both internal and external parasites. They're the go-to for sarcoptic mange. Subcutaneous injection is just more practical for herds. And again, doramectin might have that edge in duration and speed of resolution for camel mange. Strategic, well-timed administration is important.
Are topical treatments like dips or sprays still used much for camels?
Yes, topical acaricides are still used, mainly to control ticks and lice on the surface. Chemicals like pyrethroids or amidanes are common, but they can be labor-intensive. Imagine trying to dip or spray a whole herd of camels. And water scarcity in arid regions can be a real limitation. Often a combination approach works best, maybe a systemic injection for the mange mites and internal parasites, plus a topical treatment for surface ticks and lice.
Right, cover all bases. What about non-drug approaches, husbandry?
Absolutely crucial as support. Good husbandry practices are key. Things like having a quarantine period for any new animals brought into the herd to check them for parasites before they mix. Environmental sanitation, regularly cleaning, and maybe decontaminating things like bedding, harnesses, rubbing posts, those fomites we mentioned, to reduce reinfestation risk. Vector control specifically for biting flies using insecticides in housing areas, managing manure where flies breed using fly traps. And finally, good nutritional support. Keeping animals in good body condition really enhances their own immune response and resilience to parasites.
Okay, makes perfect sense for animals under direct care. But now, wild deer, you can't round them up for injections or dips. How on earth do you manage their parasites at a population level, especially when the main goal is reducing human disease risk?
Exactly. Individual treatment is usually off the table, so the strategies have to operate at the landscape and population level, aiming to reduce zoonotic disease transmission. Habitat management is one key strategy. Can you alter the environment to make it less friendly for ectoparasites, particularly ticks?
How would you do that?
Things like regularly mowing tall grasses along paths or near homes, clearing brush, removing leaf litter. These actions reduce those moist, shaded microhabitats that ticks need to survive when they're off a host. Creating barrier strips, maybe using wood chips or gravel between wooded areas and lawns, can help limit tick migration into areas where people are active. Even removing certain invasive plant species can help, as some studies show they can actually foster higher tick densities than native vegetation.
Interesting. Modifying the landscape. What about managing the deer themselves, since they're the tick amplifiers?
Yes, host population management is critical because host density drives parasite abundance. Since white-tailed deer are the essential reproductive hosts for the adult black-legged tick, their population density is strongly linked to tick abundance and Lyme disease risk. Therefore, reducing deer populations down to certain target densities, some studies suggest densities below about 8 per square mile or 20 per square kilometer through regulated hunting, is considered a primary tool for long-term tick management in many areas.
So fewer deer means fewer adult ticks get fed, fewer eggs get laid, breaking the cycle.
That's the principle, yes. Limiting the number of available hosts for those adult ticks to feed and reproduce on.
That's a direct approach on the deer population. Are there any more sort of high-tech ways to get parasite control to the deer in the wild?
There are. These are often called host-targeted technologies. The idea is to use the host animal itself to deliver an acaricide, a tick-killing chemical, to the tick population. The four-poster device is probably the best-known example.
The four-poster, what's that?
It's basically a bait station, usually filled with corn, that attracts deer. As the deer feeds, it has to rub its head, neck, and ears against rollers that are treated with an acaricide, typically permethrin. Since the head and neck are where adult ticks preferentially attach, the deer essentially treats itself.
Clever. Does it work?
Efficacy has been variable. Some studies, under ideal conditions, have shown really impressive reductions in free-living tick populations, like 92 to 98% over several years. But other studies have found more limited success. And there can be drawbacks. Concentrating deer around bait stations can lead to overbrowsing nearby, potentially spread other deer diseases. And it might not work well if there's lots of natural food available, so deer don't use the stations as much.
Right, potential unintended consequences.
Yeah.
What about targeting the other end of the cycle, the rodents that carry the pathogens?
Excellent point. Yes, there are rodent-targeted devices designed to do just that. Since small rodents are the primary reservoirs for many key tick-borne pathogens, targeting them can interrupt transmission to the ticks in the first place. One method uses tick tubes. These are simple cardboard tubes filled with cotton bedding material that's been treated with permethrin.
And the mice use the cotton for nests.
Exactly. Mice and other small rodents collect the treated cotton for their nests. The acaricide then kills ticks that are on the rodents or in their burrows, reducing the number of infected larval ticks developing. Another approach uses bait boxes that attract rodents to feed on a bait block while simultaneously applying a small dose of a topical acaricide to their fur.
Fascinating. Attacking the problem from multiple angles. What a journey this has been. Seriously, from these tiny burrowing mites causing absolute havoc in camel herds to the incredibly complex life cycles of ticks on wild deer impacting public health.
It really covers a huge spectrum, doesn't it? We've dug into the core differences, not just the parasites themselves, but their life cycles, the way they transmit diseases. That mechanical versus biological distinction is so key, and then these vastly different approaches needed for diagnosis and management.
So bringing it all together, what does this really mean for us, the listeners? How do these seemingly separate worlds connect?
Well, it perfectly illustrates the One Health concept, doesn't it? These distinct management practices, whether for camels or deer, are ultimately united by the understanding that animal health, human health, and environmental health are all inextricably linked. For the managed camelids, it's about strategic, evidence-based use of treatments combined with really good biosecurity, sanitation, and nutritional support to protect both the animals and the livelihoods that depend on them. For the wild cervids and public health, it has to be an ecosystem-level approach. You need landscape management to reduce tick habitat, maybe regulated hunting to manage deer populations, potentially careful use of those host-targeted technologies. The ultimate goal isn't necessarily eradication, that's often impossible, but reducing parasite populations and disease transmission to a level where the risk, particularly the zoonotic risk to humans, is minimized.
It's just so clear that these invisible battles fought by mites and ticks and flies on animals, both domestic and wild, have incredibly real-world consequences for all of us. So maybe the final thought for you listening is this. Whether it's a burrowing mite disrupting a pastoralist family's livelihood through their camels, or a tiny tick on a wild deer potentially increasing your risk of Lyme disease in your own backyard, the connections are undeniable. Now that you know more about these intricate links in the world of ectoparasites, what steps might you think about in your local environment, or what questions will you ask next?